Welcome to Học Tiếng Anh với Joseph, where we build vocabulary and practice pronunciation through reading. Continuing with The Cat in the Box, A History of Science in 100 Experiments, today we will read story number 73. Hôm nay chúng ta sẽ đọc câu chuyện số 73, Probing Proteins. The way improvements in technology led to advances in science is highlighted by the way X-ray crystallography was applied to the study of biological molecules. The first biomolecules to be investigated in this way were the proteins, complex molecules that were thought in the 1930s to be the key carriers of biological information, as well as providing structural material for the body. Proteins are long chains made up of subunits called amino acids. All proteins in living things on Earth are made up of different combinations of just a score of these units, arranged in different ways. Some idea of their complexity is provided by the fact that while weights of amino acids are typically about 100 units on a scale where an atom of hydrogen has one mass unit, molecular weights of proteins range from few thousand to several million units. It was crystallography that would eventually reveal the key feature of these long chain molecules is that they fold up into complex shapes, and these shapes determine their biological properties. The first steps toward this understanding were taken by J.D. vNo and his colleagues in Cambridge in 1934. vNo had worked with William Bragg in the 1920s and cut his scientific teeth by using X-ray crystallography to determine the structures of graphite and bronze. At the Cavendish laboratory, he turned his attention to organic molecules, but ran into problems with proteins because when these were dried out for study, their structure collapsed, like a structured house of cards collapsing into a disordered heap. The standard way of preparing crystals is to grow them in a concentrated solution in water, known as the mother liquor. To crystallize a protein, the purified protein is allowed to settle out of such a concentrated solution. The individual protein molecules align themselves in a repeating series of unit cells with a regular pattern, forming a crystalline lattice. John Philpott, a researcher based in Uppsala, Sweden, was doing just this with the protein pepsin in the mid-1930s. Pepsin is a digestive enzyme that breaks down other proteins in our food. When he went on a skiing holiday, he left some crystals in their mother liquor in the refrigerator in his lab, and when he got back, was surprised to find how much they had grown. A visitor from Cambridge, Glenn Milliken, took some of these crystals, still in a tube containing the mother liquor, back to Cavendish with him in his coat pocket and gave them to Bernal. Working with Dorothy Crawford, who later married and became known as Dorothy Crawford Hodgkin, Bernal first studied a crystal exposed to air while being irradiated, but found no diffraction. He had, however, also been monitoring the crystal through a microscope using polarized light. When the crystal was fresh and damp, this showed a feature known as birefringence, which indicates an ordered crystal structure, but birefringence disappeared as the crystal dried out. So they carried out more experiments with both the crystal and its mother liquor sealed inside a thin-walled glass capillary tube. This provided the first X-ray diffraction photograph of a single pepsin crystals, still in their mother liquor, in 1934. vNo's sealed capillary technique became the standard way to collect diffraction data on large molecules of this kind for the next 50 years. Bernal realized that photographs like this might be interpreted to reveal the structure of the protein molecules themselves. As Bernal and Crawford said when describing their experiment in the journal Nature, now that a crystalline protein has been made to give X-ray photographs, it is clear that we have the means of checking them, and by examining the structure of all crystalline proteins, arriving at far more detailed conclusions about protein structure than previous physical or chemical methods have been able to give. Dorothy Crawford Hodgkin went on to play a major role in the development of the application of X-ray diffraction crystallography to the study of biologically important molecules over the next few decades. But this was an immensely difficult task for reasons that slowly became clear in the years that followed. What is called the primary structure of the protein is the order of the amino acid along the chain. These chains can then be twisted around to make, for example, a helix, which is the secondary structure. And then the helix is twisted into a kind of knot in 3 dimensions, the tertiary structure. By 1971, only 7 proteins had their structure fully worked out. Since then, improving technology, including powerful computers, has led to the detailed structural analysis of more than 30,000 proteins. Moving on to the vocabulary section. Amino acids, eventually, eventually, disorder, order, disorder, purified, pure, impure, purify, align themselves, digestive enzyme, irradiated, polarized light, birefringence, capillary tube, twisted around, knot. Notice we don't pronounce the K, mình không có đọc, không có phát âm chữ K. Knot. Tertiary structure, secondary, tertiary structure. Thank you so much for your time, and as usual, I will see you in the next story. Bye for now.